a brief introduction of. Let's start it with a brief introduction of Professor Wang. Now, Professor Wang is a distinguished professor in Zhejiang University. He's the associate dean and the director of the Center for Infection and Immunity, Zhejiang University School of Medicine. The overarching goal of Professor Wang's research is to understand how nutrient acquisition, metabolic pathways, as well as different metabolites work together to shape phenotypic diversity of innate immune cells in diverse physiological and pathological settings. And this context, including inflammation, tissue repairing, and tumor microenvironments. So let's welcome Professor Wang, please.
Okay. So hello, everyone. So first, I will thank Dr. Wu Chuan for inviting me to join this fantastic online platform for the communication of immunologists during such a pandemic period. And I will also thank Dr. Chen for the nice introduction and organization. So today, I will talk about a story about another control of immunometabolism. So as a major research part in my lab in immunometabolism, and you can also get it from the name of my lab, Immunometabolism Lab. So for short, we call it MLab. Okay, so before talking the story, I hope to briefly introduce the overall research interest in our lab. So actually, the overarching goal of the research is to understand how the nutrition acquisition and also the metabolic pathways, and as well as diverse metabolites work together to shape the phenotypic diversity of innate immune cells, including tissue-resident macrophages and epithelial cells in many different physiological and pathological settings, and also in some different organs, such as the muscle, gut, and also the heart. So these contexts include the metabolic homeostasis at both system and organ levels, and also the inflammation, including acute and chronic inflammation, and the tissue homeostasis and repair. And also recently, we also enter into the field of tumor microenvironment. Okay, so today, I will mainly focus on a study investigating the role of Ionic regulation in the macrophage-mediated inflammation. So during the past several years, about recent 10 years, the immunometabolism research at the cellular level, particularly in macrophages and T cells, was greatly improved by several pioneer scientists, such as Professor Luc O'Neill, Eric Pierce, and Edward Pierce. Now, we have known that the precise regulation of macrophage activation is essential for the control of inflammatory disease. And bacterial stimuli, such as LPS-stimulated inflammatory macrophages, typically feature enhanced glycolysis, just as the Weinberg effect in tumor cells, which not only produce ATP as energy to sustain their high secretory or the phagocytic functions, but also feeds the anabolic metabolism required for their inflammatory functions. In addition, in M2, we call it M2-like macrophages, the TCA cycle is intact and participate in oxidized phosphorylation and providing ATP for energy. And the glutamine metabolism and also the TCA metabolite alpha Kg are also critical for M2-like functions. And in M1-like macrophages, the TCA cycle is broken in two places. One is the citrate, the other is succinate. And citrate is usually used to generate fatty acids for membrane biogenesis and also for the prostaglandin production. And the TCA cycle in M1-like macrophages also generates the ataconic acid, which has the direct antimicrobial activity via the enzyme IRG1. So our group has previously identified adequate supplies of essential amino acids, such as the serine and the methionine, as being critically involved in the induction of inflammatory cytokines, such as the interleukin-1. And we found that LPS activates the PPP pathway, which is the pentose phosphate pathway, the SSP pathway, which is the serine synthesis pathway, and the 1-carbon metabolism. So we call it SGOC metabolism network. And this network can synergistically drive epigenetic reprogramming for L1 beta expression. And we found the glucose-derived ribose and the 1-carbon units fed by both glucose and extracellular serine are synergistically integrated into the methionine cycle through the de novo ATP synthesis and fuel the generation of a very important metabolic metabolite, SAM, during the LPS stimulation. And we found that the SAM generation support mainly the histone H3 lysine 36 trimethylation for L1 beta production. So we therefore identify a synergistic effect of glucose and amino acid metabolism on the production of SAM, which is essential for macrophage-mediated inflammation. So to acquire constant extracellular nutrients, the rapidly proliferating cells, such as tumor cells, import nutrients that both fuel the ATP generation and also the synthesis of the building blocks for new cells. So as strategies used for nutrient uptake, the tumor cells usually can upregulate the glucose and glutamine transporter genes, and as well as prevent their degradation through the lysosome pathway to support their huge demand of anabolic metabolism. As just I mentioned, although anabolic metabolism is considered as a hallmark of cancer, many of these metabolic changes also occur in inflammatory macrophages. For example, the glycolysis and the amino acid metabolism. But however, macrophages are usually not prone to rapid proliferation upon inflammatory activation, such as LPS. So how macrophages acquire the constant supply of extracellular nutrients to feed inflammation is the scientific question of this study. And so I want to introduce a very important concept, which is the membrane potential. The negative plasma membrane potential generated by different ionic gradients across the membrane is a key electrochemical aspect of cells. Precise control of membrane potential is essential for the physiological functions of excitable cells, such as neurons and cardiomyocytes. Interestingly, membrane potential has also recently been linked to cell survival and the proliferation triggered by mitogenic signaling in cancer cells and also the neurons. So this data suggests that this old electrochemical characters has broader functions. And however, as non-excitable cells, because the immune cells are usually non-excitable, so the function of membrane potential in immune cells, particularly in macrophages, upon inflammatory stimuli are still largely unknown. So ion channels and transporters mediate transport of charged ions across the hydrophobic lipid membranes. Although immune cells are usually non-excitable cells, they also express ion channels and transporters to regulate the membrane potential and the cell signaling, as well as the physiological cell functions, such as the gene expression, cell death, apoptosis, proliferation, development, and migration. In macrophages, divalent cations such as the calcium, magnesium, and zinc have important roles as second messengers to regulate the intracellular signaling pathways. By contrast, the contributions of the monovalent cations, such as the potassium efflux, sodium influx, and the chloride efflux, they together determine the membrane potential, which indirectly control the influx of calcium and also the immune cell signaling. Regarding to the membrane potential, the resting macrophage membrane potential is usually around -50 millivolt. So if the membrane potential is more positive than it is at the resting membrane potential, for example, it is the -30 millivolt, we call it depolarization of the membrane potential. In contrast, if the membrane potential is more negative than it is at the resting membrane potential, we call it hyperpolarization. Okay, so first we want to explore the impact of the membrane potential on inflammatory macrophages. So we manipulated the membrane potential of LPS stimulated peritoneal macrophages, measured by the whole cell patch clamping or the membrane potential indicator dye by increasing the extracellular potassium or applying gramazine, which are two widely used methods for the membrane potential depolarization, but through distinct mechanisms. We also made sure that the other method was used when the viability of macrophages was not affected by measuring cell death, such as the apoptosis or the lytic cell death, and to seek the bona fide changes induced by the membrane potential depolarization, we perform combined analysis with RNA-seq data from each the strategy to the depolarize the macrophages, and we found a strong correlation between both treatments in terms of their impact on the transcriptome. And remarkably, when we analyze the shared differentially expressed genes between the two treatments, the common downregulated genes were particularly enriched in the inflammatory response and the defense response, indicating that there is anti-inflammatory effect of the membrane potential depolarization in activated macrophages. By contrast, despite adequate extracellular nutrient resources, the common upregulated genes were mostly enriched in pathways typically induced under nutrient restriction, including response to starvation, autophagy, and the catabolic process. And we also use the GSEA analysis to analyze the RNA-seq data and found the very significant enrichment in autophagy, response to starvation, response to GCN2, to animal acid deficiency, and animal acid transport in the settings of the elevated extracellular potassium and gramazine. So to further determine whether these changed transcriptional programs were indeed induced by nutrient restriction, we performed unbiased metabolomics profiling. And although the elevated extracellular potassium and gramazine showed some difference in changed metabolites, we found a substantial proportion of metabolites similarly altered by these two strategies. And using the metabolic pathway enrichment analysis, the common decreased metabolites were mainly metabolites associated with glycolysis, PPP pathway, TCA cycle, purine synthesis, phospholipid biosynthesis, and animal acid metabolism and biosynthesis. So therefore, these changes in metabolites suggest an impaired nutrient acquisition and a subsequent anabolic metabolism upon membrane potential depolarization. And we found a very profound impact of the membrane potential depolarization on the immunometabolism of the macrophage upon inflammatory stimulation. So next, although these data indicate a critical role of the membrane potential in macrophage, we need to identify a physiological modulator controlling this memory potential dependent regulation of inflammatory macrophages. And because given the key roles of the potassium conductance in governing the resting memory potential, we compare the expression of 54 potassium channel genes in BMDMs using RNA-seq, and we found several highly expressed potassium channels. Among them, we focused on inwardly rectifying potassium channel KIR2.1, which mainly play roles in regulating the memory potential in the excitable cells, such as the cardiomyocytes and the smooth muscle cells. But there's no study of this channel in inflammation and also in the immune cells. So we made the whole cell PET clamp recording. So this the results. We use this strategy to measure the memory potential in the primary macrophages. And we also use the ML133 is a widely used selective KIR2.1 inhibitor. And also we use the mice with a specific deletion of KIR2.1 in myeloid cells. So we recorded a typical KIR2.1 current and found that ML133 potently inhibits this KIR2.1 currents in both resting and the inflammatory macrophages. Consistently, the KIR2.1 current was also abolished in the knockout mice and knockout macrophages without affecting their differentiation. And the most important, we found a depolarized memory potential in both the KIR2.1 deficient, resting, and activated macrophages. And we also found that the ML133 depolarized the memory potential in a dose-dependent manner. And we found that the 25 micromolar ML133 led to a memory potential depolarization comparable to that induced by the genetic deficiency. So we choose this dose of the 25 micromolar in most of the subsequent experiments. Okay, so because the major function of inflammatory macrophage is to elicit inflammation for host defense. So we use GSEA of RNA-seq data from the LPS stimulated BMDMs and demonstrate a very striking enrichment of the inflammatory response after ML133 treatment. So this data just recapitulated the anti-inflammatory effect of the memory potential depolarization by the above two strategies in macrophages. So further analysis revealed that a cluster of inflammatory genes, including L1 beta, L1 alpha, L6, L18, L12, and 6L10, they were suppressed by ML133, while another very important inflammatory gene, TNF alpha, was not affected. And consistently, we found a similar reduction of L1 beta and the L1 alpha transcription by either the ML133 or the KIR2.1 deficiency with no change of the TNF alpha production. And in addition, the absence of additional effects of ML133 in the knockout macrophages further confirmed the specificity of KIR2.1. And also, you can see that the elevated extracellular potassium or the gram spin showed a similar suppression of these genes. So that means that is a general effect of the memory depolarization on this cluster of inflammatory genes. Because we didn't find the change of the TNF alpha production, so we hypothesized that the KIR2.1 will not be required for the general signaling pathway essential for most LPS-induced genes, such as the TOR4 signaling. Indeed, we found that the LPS-induced NF-kappa B and MAP kinase activation was not affected. So we next consider the possibility that KIR 2.1 promotes inflammation by modulating the calcium-dependent signaling, because the potassium-mediated memory potential can indirectly affect calcium flux. However, the ML133-induced L1 beta suppression was independent of the concentrations of extracellular calcium or the other divalent cations. And in addition, when the extracellular calcium was chelated by BAPTA or EGTA, the effect of ML133 was not affected. And interestingly, in the setting of chelating the intracellular calcium by BAPTA-AM, we found that this treatment alone was sufficient to suppress L1 beta and L1 alpha, and had an additional effect on the basis of ML133. And when we depleted intracellular calcium stores using TG, ML133 showed a similar inhibitory efficiency on L1 beta production in the presence or absence of TG. So this data suggests a separate mechanism of KIR 2.1 independent of the intracellular calcium. As mentioned above, that we have previously reported a syndrome of glycolytic off-source and amino acid metabolism driving the expression of a cluster of inflammatory genes, and including L1 beta, L6, L18, L12, L27, and 6CL10, but not TNF alpha. So surprisingly, this suppression of this cluster of metabolically responsive genes was perfectly recapitulated by ML133, suggesting that a metabolic mechanism underlies the regulation of KIR 2.1 in inflammatory macrophages. So consistent with this idea, we used unbiased metabolomics profiling of LPS-stimulated macrophages, and we revealed that the decreased metabolites represented anabolic metabolism after ML133 treatment, just like the change of the metabolites affected by increased extracellular potassium or the gram stain. Both unbiased metabolomics profiling, and we also used a targeted metabolomics approach. They both reveal a significant reduction of LPS-induced glycolysis intermediates by ML133. So this was further validated by the decreased ECAR. We used ECAR to measure the glycolysis efficiency. And we also found that the metabolites R5P and 3PAS. So these two metabolites represent the glycolytic off-source, including the PPP pathway and the SSP pathway, and they are all decreased by ML133. And also, the amino acid essential for the metabolism responsive inflammatory genes, including the serine, glycine, methylene, they were all decreased by ML133. So why? So we found that these metabolic alterations were led by a reduction in electrochemically dependent nutrient uptake, because we found that the macrophage with depolarized memory potential due to ML133 or its deficiency had diminished LPS-induced glucose uptake, which was also recapitulated by gram stain-induced memory potential dehydration. And the similar results were also obtained in the knockout mice by measuring the LPS-induced glucose uptake of the peritoneal exudate cells and the inflammatory monocytes in vivo. By assessing the carbon-labeled serine and the glycine derived from the carbon-13 glucose tracer, we found that SSP field by glucose uptake was impaired upon memory potential depolarization by KIR2.1 repression, and also by the elevated extracellular potassium or gramsci. In addition, unlabeled serine, glycine, and methionine, which will be the N+0, they were all decreased in these activated macrophages after the memory potential depolarization. So this data together indicate a profound impact of the KIR2.1 mediate the memory potential on nutrient acquisition of macrophages upon activation. And the GSEA of RNA-seq also highlighted the significant enrichment in the amino acid transport and the SGOC metabolism. And we further analyzed the differentially expressed genes shared between ML133 and the KIR2.1 genetic deficiency. And we found that besides the common downregulated genes mostly enriched in inflammatory response and the response to LPS, so this well reflected the anti-inflammatory effect of the KIR2.1 repression. More importantly, we found the common upregulated genes were particularly enriched in pathways, including in response to starvation. So this data suggests a critical role of KIR2.1 in nutrient acquisition to feed the metabolic underpinnings of inflammation. The impaired PPP pathway and SFP pathway fueled by glucose uptake and decreased serine and methionine uptake. So we want to, because this data, we want to quantify a very important critical epigenetic metabolite, SAM, because its synthesis is synergistically fed by these metabolic inputs. So we found that either ML133 treatment or KIR2.1 deficiency led to a significant reduction in the cellular SAM of LPS-stimulated macrophages. When we used the carbon-13 glucose chasers, we measured a reduction of LPS-induced M+5 to 9 SAM. So this mainly from the aware both the PPP pathway and the SSP pathway, and also they are precursors of the M+5 to 9 ATP in the setting of memory potential polarization with depolarization with ML133, it elevated extracellular potassium and also the gramsci. So we next also examine the SAM generation fueled by not only the glucose, but also the amino acid uptake, such as the serine. So we've also found a similar reduction of the M+1 to 4 SAM fed by the carbon-13 serine derived carbons. So to determine whether this association was causal, we supplemented ML133 treated inflammatory macrophages with SAM. And we found that the suppressed L1 beta mRNA production was dose-dependently restored. And the limited nutrient uptake and subsequent caloric restriction also led us to quantify acetyl-CoA, because acetyl-CoA is also important. It can enable carbons derived from diverse nutrients to be utilized in the TCA cycle for the oxidative phosphorylation. And also it can serve as a metabolic substrate for epigenetic regulation. Yes, we indeed found LPS-induced acetyl-CoA was significantly decreased by ML133. But when we supplemented ML133 treated inflammatory macrophages with acetate, which is the immediate precursor of acetyl-CoA, we found that L1 beta and L1 alpha production was not restored. And we also detect another important epigenetic metabolite, the TCA cycle intermediate alpha Kg, which is also a very important epigenetic regulator. And we also found no rescue effect on the L1 beta production. So this result suggests that a greater dependence of the memory potential induced inflammatory program on the epigenetic programs fueled by SAM generation. And to assess the epigenetic consequence of the limited SAM generation, we analyzed the expression of about 200 annotated SAM-dependent methyltransferase enzymes, and we found a remarkable upregulation of all methyltransferase for histone 3 lysine 36 methylation compared to those for other methylation marks. And among them, ASH1L and NSD, they had the specific mono and dimethylase activity for H3K36. And SATD2 is the only reported trimethylase catalyzing the trimethylation of H3K36. And also, because the main distribution of H3K36 trimethylation is usually in a wide range of the gene body regions, so we call it a SAM sink. And so, because this, it makes us to think it is reasonable for the H3K36 methylation is more sensitive to SAM production for gene expression. So, consistent with this idea, we use the cheap qPCR, and we found that memory depolarization by KIR 2.1 repression or elevated the extracellular potassium, they can decrease the LPS-induced H3K36 trimethylase occupancy in L1 beta gene body regions. And we also found similar results of other metabolism responsive genes, including the L1 alpha, L18, and the CXL10. So this data suggests that KIR 2.1 repression leads to the depletion of intracellular SAM pool, and this SAM is critical to fill the histone methylation marks, at least include the H3K36 trimethylation, and thereby they limit the epigenetic imprints of these metabolism responsive inflammatory genes. What's the mechanism? We speculate that there would be common changes in the gene transcripts representing a specific mechanism underlying the memory potential control of immunometabolism. Therefore, we perform combined analysis with RNA-seq data from LPS-accumulated macrophages with ML133, gramstine, or the elevated extracellular potassium. We found more than 1,000 differentially expressed genes shared by all three treatments. As expected, the common regulated genes were mostly raised in the inflammatory response, and common upregulated genes were enriched in the key pathways of metabolic adaptations in response to nutrient starvation, such as autophagy, amino acid transport, and the negative regulator of mTOR signaling. And remarkably, we noted that the upregulation of a set of the solute carrier SLC family members, including the 4F2 heavy chain. So this is also known as CD98 or SLC3A2. And this is also recognized as a critical player in nutrient uptake. So 4F2HC is not itself a nutrient transporter, but it can act as a chaperone that interact with different light chains to form a stable heterodimer transporter for the uptake of many amino acids, as well as facilitating the stabilization of the glucose transporter 1, GLUT1. So GLUT1 is the primary glucose transporter in macrophages. So we consider whether the KIR 2.1 mediated memory potential supported the nutrient acquisition to fill inflammation by modulating the nutrient transporters. So to examine this hypothesis, we used the flow cytometry, and we detected the induction of surface expression of both 4F2HC and GLUT1 upon LPS stimulation. This was dose-dependently inhibited by ML133 and similarly by the elevated extracellular potassium or gramstine. So the decreased surface expression of GLUT1, we also used another strategy. We used the immunoblot for biolated cell surface proteins. And we found that the impacted nutrient uptake of macrophages with a depolarized membrane potential is possibly because of the loss of the critical nutrient transporters on the cell surface. And to gain a deeper understanding of the impact of membrane potential depolarization on the molecular events occurring around the cell surface, we extract the plasma membrane proteins of LPS-stimulated macrophages for further the quantitative proteomics analysis. And we found that more than 200 differentially expressed proteins was shared between these different treatments, such as the ML133 and the elevated potassium. And we found these proteins are mainly enriched in the memory trafficking, including the vesicle-mediated transport, endocytosis, and the phagocytosis. And this data suggests a contribution of endocytosis to the depolarized membrane potential-induced loss of cell surface nutrient transporters. And this is also consistent with the view that the nutrient uptake is usually tightly controlled by the dynamic memory trafficking of such transporters. We indeed found that after ML133 or extracellular potassium treatment, LPS-stimulated macrophages showed significantly enhanced internalization of both GLUT1 and 4F2HC. And we also used the endocytosis inhibitor now study, and we found it can partly restore the decreased levels of surface GLUT1, glucose uptake, and L1 beta production. And because the GLUT1 and the 4F2HC are the ARF6 GRP1 cargo proteins, so that means they usually can be recycled back to the plasma membrane via the tubular recycling enzyme. So we use a constitutively active mutant of GRP1, which can force the recycle of these transporters back to the plasma membrane and prevent their loss. And we found it can largely restore the production of L1 beta. And essentially, a depolarized membrane potential leads to changes in the electrochemical properties of the plasma membrane, which is composed mainly of charged phospholipids. In particular, PIP2 enrichment in plasma membrane microdomains plays a central role in membrane trafficking and endocytosis by triggering the plasma membrane invaginations and recruiting endocytic factors. Surprisingly, so we found that PIP2 enrichment in the because PIP2 enrichment in the plasma membrane microdomains play a central role in the memory trafficking and endocytosis. So we use the PIP2 antibody, and we found that although the amount of the plasma membrane PIP2 was not changed, but we found that the ML133 or elevated potassium can lead to significantly enriched PIP2 clustering on the plasma membrane of the LPS-stimulated macrophages. And more importantly, the ML133 induced decrease in glucose uptake and L1 beta production can be restored by expression of the lipid phosphatase score B, which is an efficient means to deplete the PIP2. So this data suggests a critical regulation of KIR 2.1 mediated membrane potential, largely by modulating the memory phospholipid dynamics for the surface retention of nutrient transporters. So this can ensure the nutrient acquisition of macrophages to fuel inflammation. Last, to explore the physiological relevance of this regulation, we examined the phenotype of inflammation in vivo. when the KIA 2.1 regulation of membrane potential was absent. So using a in vivo LPS-induced sepsis model, we measured decrease in serum levels of L1 beta, L1 alpha, and L6, as well as increased the survival of mice, and this effect was similarly found in gram-negative bacterial infection-induced inflammation, both in vitro and in vivo. And in human samples, the RNA-seq data from the blood monocytes of patients during sepsis and after recovery showed that the KIA 2.1 was remarkably upregulated in monocytes from sepsis patients compared to healthy donors. And in addition, the KIA 2.1 was more strongly increased by LPS stimulation in monocytes from healthy and recovering donors, which are indicative of an endotoxin sensitive phenotype than that in monocytes from septic donors, which are indicative of endotoxin tolerant phenotype. And strikingly, there was similar patterns of the cluster of metabolism responsive inflammatory genes, including L1 and L6. And last, we used synovial fluid cells from GOTI patients, which usually suffer from by the production of L1 beta. And we found that the ML133 can suppress L1 beta production in these freshly isolated synovial fluid cells from GOTI patients. So this suggests a potential anti-inflammatory suggestion may be targeted by KIA 2.1. So in this work, we found that the plasma memory potential of macrophages mediated by KIA 2.1, which is a inwardly rectifying potassium channel, is a critical determinant of nutrient acquisition and the subsequent metabolic reprogramming driving inflammation. In the absence of KIA 2.1 activity, it will lead to the depolarization of the macrophages and further lead to a caloric restriction state by limiting the nutrient uptake. And it will lead to the consequence of the depletion of the epigenetic substrates, feeding the histone methylation at the loci of a cluster of metabolism responsive inflammatory genes, including the L1 beta. And we also found that the KIA 2.1 mediated membrane potential supports the nutrient uptake by supporting the cell surface retention of key nutrient transporters such as the 4F2HC and GLUT1 by its configuration of plasma memory phospholipid dynamics. So therefore, we propose an Ionic and memory potential control of nutrient acquisition. Use this strategy, the inflammatory macrophages can acquire a constant supply of the molecular building blocks to support their inflammatory functions. Okay, so that will be the end of my talk today, and I will thank my collaborator, Dr. Yang Wei, and all my talent students, and in particular, Doctors Yu Wenwei, Wang Zhen, and Yu Xiafei, and all fundings that support my lab. And thank you all very much, and I will be happy to take any questions.
Okay, thank you for the wonderful talk. So is there any questions from the audience? I will start with my two questions. So first of all, I think it's amazing that the KIR 2.1 expression was highly upregulated after LPS stimulation, right? So what is the mechanism for that? What is the signaling parts of the LPS would to induce the expression of KIR 2.1? Second one, in the patient's samples, I saw that when you're using the KIR 2.1 inhibitors, would inhibit the LPS-induced inflammation in the clinical samples. So I think it's very interesting that so long as the KIR 2.1 is not affecting the toll-like receptor 4 receptors, right? And the signaling, right? So why the inhibition of KIR 2.4 will reversely inhibit the LPS signaling? Okay.
Okay, so as I just mentioned that the KIR 2.1 function is not dependent on its regulation of the TOL4 signaling, the general signaling for the production of inflammatory cytokines, because you can see that this regulation don't affect the production of TNF alpha, because usually the TNF alpha and L1 beta, they all rely on the signaling introduction of such as TOL4 signaling, but we found a very different effect on between the TNF alpha and L1 beta. We have no effect on TNF alpha production. And according to our previous study, so we focus on the metabolic, immunometabolic mechanism for this KIR 2.1 regulation. So the detailed mechanism is because the KIR 2.1 expression is critical for maintaining the memory potential of macrophages, including inflammatory macrophages. Without KIR 2.1, the macrophages will be depolarization. So because the depolarization, that will induce the dynamic exchange of the memory, the plasma memory phospholipid, including the PIP2, and then it will inducing the enhanced endocytosis of very key nutrient transporters, including the amino acid transporter and also the glucose transporter, GLUT1. So if without this KIR 2.1, this nutrient transporter will largely enhanced by endocytosis and loss on the cell surface, then the macrophages cannot acquire a constant extracellular nutrient supply to feed the inflammation, because these nutrients are critical to feed the generation of a very important epigenetic regulator, SAM. And the SAM can regulate the expression of L1 beta, L1 alpha, and L6 expression on the epigenetic level. So that's the mechanism of this study. Yeah, and about the second question you asked, why it can suppress the inflammation in the human patients. We use the GOTE patient. It's not induced by the bacterial infection, but induced by the DAMP induced. It's a kind of steroid inflammation, usually caused by the uric acid. The excess uric acid which can induce the L1 beta productions through through L1 beta expression and also through the inflammation dependent processing of L1 beta production. So you use this patient samples, we found that the KIR 2.1 suppression can suppress the L1 beta production, and based on our model, it mainly suppressed the L1 beta transcription on the transcription level. Yeah.
Okay, thanks. There's a question from Liang Chi. Wonderful talk. Do you measure the ROS level in macrophages, and does the activated one carbon metabolism pathways reduce the macrophage produced ROS?
Yeah, that's a good question. In our previous study, we found that the PPP pathway usually can produce NADPH, which is a key controller to control the excess RS in the macrophages. But if you block the PPP pathway, it will decrease the NADPH synthesis, and then it will induce the RS production. But it not will but in this setting, the elevated RS production is not benefit for the production of inflammation, but it's not good for the inflammation production, because excess RS production can also make the inflammatory macrophage stress and unhappy. So it will decrease the production of the inflammatory cytokine. So the RS production is not always benefit for the inflammation. It depends on the dose and also the location, because it will the mitochondrial RS or the cytoplasma RS. Yeah, but in this study, we exclude the contribution of RS. We have measured that.
Okay. Is there any other question from the audience? Okay.
Hey, Wang. Thanks for the talk. It's great. I have questions about I still have the question about why LPS can upregulate the KIR 2.1.
Oh, sorry.
Why LPS can upregulate the channel?
Yeah. It's I didn't show the expression change of
You
KIR
did
2.1.
I think in your sequencing data.
Yeah. Actually, less than 6 hours after LPS stimulation, the mRNA and also the protein expression of KIR 2.1 will be increased by LPS stimulation. But after that, we found a dramatic decrease of KIR 2.1 expression. I didn't mention in this here, because this study mainly focused on the acute induction of inflammatory stage. So we just used the macrophage stimulated by 6 hours of LPS. So your questions, why LPS induced the induction of KIR 2.1 expression?
Yeah.
Yeah, we didn't put too much attention on this because it has function. But even the expression of KIR 2.1 expression increased by LPS, we found a stable memory potential during the acute stage of LPS-induced inflammatory macrophage. So which means even though the expression increased, but the function is stable. It can maintain a very stable memory potential of macrophages. But if you block or deficient this channel, it will dramatically induce the depolarization of the memory potential. So we think that the expression level didn't contribute too much on this regulation.
Well, the potassium channel problem is if you block one, they actually depolarize everything. It doesn't matter if it's KIR 2.1 or KCN 1.2, KV 1.3. I mean, the problem is the block of the potassium channel, any of these channels, can induce membrane potential change. And then you also show, so I'm just not sure that how specific this KIR 2.1 function in terms of the phenotype of LPS stimulation. But regardless, my other question is, you show that when you actually depolarize or you stimulate the macrophage with the LPS, you find increase of intracellular calcium increase, which probably come from ER, right?
No, I didn't mention that. We exclude, the part of calcium, we just exclude the contribution of calcium. We have evidence that the intracellular calcium release can independently affect the L1 beta production.
I know. That's not my question. My question is, when you actually depolarize the macrophage, you find the increase of ER release calcium. Is that correct?
I didn't show the data. Really? In this.
I saw you using the chelation BMPM-AM, BMPT-AM.
Yeah, that's only for chelating the calcium.
I know, but did you find increase of?
Yes, we have the data. We have it. If you block the KIR 2.1, it will induce the calcium release. But I didn't say it's here, because the data I show is just to exclude the calcium contribution to the KIR 2.1 regulation. So we used the chylation, the both extracellular calcium and intracellular calcium, but we found that there's no effect. There are no effect on the function of KIR 2.1. And also we use different medium with different concentrations of calcium, extracellular calcium, and we found that the effect of KIR 2.1 didn't change. So we conclude that the regulation of KIR 2.1 is independent of the calcium signaling. But indeed, we found the intracellular calcium released from the ER have effect on L1 beta production, but that regulation is in parallel to the regulation of KIR 2.1. Yeah.
Okay, so the other thing is you say the KIR 2.1, the deletion changed the phospholipid, therefore the GLUT1 expression is decreased. That's what you say?
Decrease from the cell surface.
Yeah, decrease from cell surface. But my question is, if the phospholipid structure changes, that's actually change everything. It shouldn't be specific towards to the GLUT1. It should actually generalize to all the receptors on the cell surface. And how can you prove this actually the depolarization is actually caused the IL-1 beta expression defect is because of the nutrient uptake?
Yeah, that's a good question. Yeah, because we use we check the plasma membrane proteins for the quantitative the proteomics analysis, and we found that all changed the proteins are mainly involved in endocytosis. Yeah, you are right, because many other receptors or transporters are also regulated by endocytosis. But in this study, we use the PIP2 depletion strategy, and it can restore. We observe the restore effect of the glucose expression on the cell surface and also the L1 beta production. So logically, we think it depends on the dynamics of the plasma phospholipid. But you are right, we didn't measure the other transporters or nutrient transporters in this study. So we just say at least is a fact, GLUT1. And also because, you know, the 4-HC or 4-2-HC is, it can interact with a lot of transporter protein to form the different heterodimers to transport different amino acids. So it not only affect the serine or methylene uptake, it can also affect the arginine and leucine uptake. So in this study, we didn't say that it's only narrowed down to the regulation of the serine or methylene. We want to say that it's a more general function of the stable memory potential of macrophages in maintaining the macrophages to acquire the constant extracellular nutrients during inflammation to feed the anabolic metabolism of the inflammatory macrophages.
Okay. I think my last question is, you actually mimic the depolarize of the KIR 2.1 with ME133. Is that the compound name? I forgot the name.
ML133, yeah.
M what?
ML133.
Yeah, ML133. My question is, if you think the depolarize is depend on this channel, have you done the knockout mice treated with ML133?
No, ML133 is the selective inhibitor. And we also use the KIR 2.1 condition knockout in myeloid cells.
No, I know.
So we use both.
Yeah, I know. I'm
I understand your question. You mean you use the ML133 in the knockout mice?
Yeah.
Yeah. We have a lot of data. So there's no additional effect on the knockout mice. So we can very confident to say that this inhibitor is very specific and selective. Yeah, because a lot of reviewers asked this question, and we supplemented a lot of experiments to determine the specificity of ML133.
This is an antagonist or agonist?
It's just a blocker.
Specific for the KIR 2.1?
Yeah, it's a selective inhibitor for KIR 2 family, not only KIR 2.1, also 2.1, 2.2, 2.3, but only 2.1 have the highest expression in macrophages, not 2.2 and 2.3. And we also use the patch clamp to exclude this inhibitor on the other expressed channel in macrophages, such as you mentioned, the KV 1.3, K calcium 3.1, KIR 7.1. We all detect that they have no off-target effect on the other channels.
Okay, thanks.
Yeah, thank you.
Hey, Wang, this is really beautiful talk, and I really enjoy it. So maybe here I first have a sort of naive question. So when we say the depolarization of the membrane, does it mean the negative charge on the inner leaflet of the plasma membrane is sort of decreased? Could I understand in that way?
Yeah. So you can say that.
So if that's the case, so basically that will affect the recruitment of many of these cytosolic protein that is depend on these negatively charged lipids on the membrane, for example, PIP2 or PS. Is that correct?
Yes.
Okay, I see. So that can sort of explain many of the trafficking phenotype. The other, I think, maybe is also kind of basic question. So when we talk about, I think, the polarization, depolymerization is mostly like I think we first learned the concept in neurons. So when neuron undergo these polarization, depolymerization, so they can like slowly relapse and come back to the sort of the steady state. So in this case, it looks like the KIR21 can have a more sustained effect. Is that, if I understand here correctly?
Yes, you're right. The immune cells, including the lymphocytes and the macrophages, they are all non-excitable cells, not like the neurons or muscle cells. So their memory potential are relatively very stable, even during the inflammation or resting stage. So, but if you knock out or block the KIR 2.1, you can dramatically form the depolarization, the increase the memory potential of these immune cells. And in these settings, the immune cells lost the ability to acquire the extracellular nutrient. So I know I can imagine, I can guess your question deeply, that because we didn't find a more physiological or pathological context to observe that maybe in a specific disease, we found the memory potential of macrophages change in vivo. But we didn't find this contact because, as I mentioned, that they all stable, they all have stable memory potential. We only show the data if we block or we change the memory potential, we can find something. But so far we didn't have the evidence to see whether it can naturally happen in vivo during some pathological context. But only maybe a context will be interesting to detect this, and this is also a work we are going to study in our lab. So I can say something a little here. So in the tumor microenvironment, there are a lot of evidence showing that there will be elevated the potassium concentration in the intratumor fluid. So which means there will be an increased extracellular potassium in the tumor microenvironment. So in this microenvironment, the increased potassium will lead to the depolarization of immune cells. and so that possibility will decrease the.
Yes. So is there any probe, let's say fluorescent-based probe, that you can measure these membrane potential?
We are working on it. We are working on it because calcium you can find some indicator or probe, but the potassium is very hard in the field. Can find a very specific indicator or later to manipulate the potassium, even in the in vivo context. So we are working on it. We hope in the future we can work on it. Yes.
Okay, that's great. So my other question about the depolarization induced the PIP2 clustering effect on the plasma membrane. So do you think that is mediated by the reorganization of these like transmembrane protein, because like PIP2 binds many of these like marks so it is a reorganization of the lipids or it's mediated by the protein reorganization on the membrane?
We didn't we have no evidence to reorganization of the protein, but we have data of the expression of the protein on the cell surface, because we extract the memory potential proteins for the quantitative proteomics analysis, and it's when we observe the PIP2 clustering, it's very obvious in our system. You can see very dramatic clustering of the PIP2. So we think maybe it is a more basic effect of the memory potential because it's an electrochemical factor. So this factor can be reasonably seen to induce the relocalization of the PIP2, because this phospholipids are very depend on the electrochemical properties. Yeah.
I see. And the PIP2 clustering can be maintained for a pretty long time after the, let's say, the potassium change.
Yeah, we didn't observe a very long time in our system. We just maybe half an hour. I don't remember the deal. Yeah,
that's actually pretty long in terms of the signaling response, because most of these like either the PIP2 hydrolysis or these reorganization usually occurs very quick. So I think half an hour is a pretty long, pretty stable effect, which is also very interesting. So my last question is like the KIR 2.1 is, I think, mostly highly expressed in neutrophils, or even much higher than in the macrophage or monocyte. So do you see like they have a similar regulatory mechanism, or if this has been explored?
Yeah. I also observed that it's highly expression in neutrophils, but we didn't detect the function of the neutrophil. But in our study, we cultured the BMDM and in vitro, so we observed the changes in macrophages. But in the in vivo data, we really cannot exclude so far based on our data, we cannot exclude the contribution of the neutrophils. Yeah.
Okay, great. Yeah, thanks so much.
Yeah, thank you. Thank you all. Yeah. So
let me check if there are any more questions. Okay, so maybe I think that's it. Okay, thank you so much, Professor Wang, for this interesting story about the macrophage inflammatory and the metabolism, and also all the questions and the responses is very wonderful. Thank you all guys. Thank you. See you next week.
Thank you all. Thank you. Bye.
Thank you. See you.
See you.
See you.